Why would we be interested in delta-8? There's some positives and negatives, so let's start with the positives. Delta-8 has been shown to have some really high potential for therapeutic and recreational applications. Scientifically, like the other minor cannabinoids, it's of significant interest. It's relatively easy to produce without a lot of large capital investment. And right now, all that production is without regulatory oversight. In addition, due to oversupply of CBD, the margins of CBD have been depressed. And so converting CBD, which is a low margin commodity, into a higher value added product, it has some financial salvation to investors who may have invested in the hemp world waiting for FDA approval of CBD. Like Delta-9 THC, the therapeutic potential is there. It's neuroprotective properties with the production of neurotransmitter acetylcholine, which is responsible for memory, cognition, and neuroplasticity. It's been shown to be effective for appetite stimulation, antiemetic effects, analgesic properties, as well as anxiolytic properties as it binds to the CB1 and CB2 receptors. There's been a lot of work that's been published in this realm. A lot of people say, well, we don't know anything about delta-8. Well, we do. It's shown to have high degree of efficacy for certain conditions. One of the studies I read was on treatment of pediatric cancer patients, and it wasn't getting them stoned and it was stimulating the appetite. On the other hand, some of the rationale against the delta-8 argument is that it has a potential for intoxication. One of the things that the hemp industry has established is that we've made some significant legislative progress on hemp, and a lot of that was based on the fact that hemp is not an intoxicating product. So now that we show that delta-8 can be derived from hemp and has some psychoactive components, is that going to stall or hurt our legislative progress? Unregulated products are also cutting into the regulated cannabis products. So you can buy delta-8 products at every convenience store now, vape store, you can buy it online. And that's taking money away from the regulated marijuana sales. When these producers are spending millions of dollars on licensing and regulatory processes, this can hurt their bottom line. And then the other issue is consumer safety. And that's really where my focus on. These products are being produced with very little regulatory oversight. There's many contaminants that we see that have not been identified, and as unidentified, we don't know their toxicity, and so that raises some significant concern. I hear all the time that delta-8 is natural. I hear that they're extracting it from the hemp plant. But in the analysis of over 18,000 samples, 98.5% of them had no measurable delta-8. For the remaining samples that did have delta-8, the average was 0.0018%, so it's extremely low trace levels of delta-8 that occur naturally. In order to extract it and produce a natural delta-8, it would take about 55,000 kilos of hemp to produce 1 kilo of delta-8. At $400 per kilo, that's about 22 million just in biomass. If you add in the cost of production, extraction, and isolation, that would mean that the estimated market value would be somewhere between $200 and $500 million per kilo, which seems a little bit impractical, especially when you consider I can go to the International Space Station for $74 million. Natural products. We talk about cannabis as a natural product, and it's not the only natural product. We know that salicylic acid was an early remedy for pain, fever, and inflammation, going back over 4,000 years. It's found in the bark of certain birch and willow trees, and so maybe 100 years ago, if you had a toothache, you might chew on this bark. But as the pharmaceutical companies started to understand what was chemically responsible, they started acetylating it and marketing it commercially as aspirin, which we're all very familiar with. Well, is that aspirin a natural product, or is it a synthetic product? I think most people would agree when we moved away from pulling it out of nature and doing the chemical process that it would be considered synthetic. So let's talk about some other synthetic chemistry. There's a very simple reaction to convert pseudoephedrine or cold medicine into methamphetamine or crystal meth. Just because the starting material is legal and has been studied for safety does not mean that the resulting product is necessarily legal or studied for safety either. And if you look at some of the ingredients or constituents that are required for that conversion, lot of those are going to be very similar to what is used for the isomerization of CBD to delta X THC. I call it delta X THC because the delta bond is oftentimes in very different positions, and you might get delta 9, you might get delta 8, you might get delta 10. You don't often have complete control of that. And so as I think about synthetic chemistry, whether it's making crystal meth or isomerizing CBD, I'm thinking about contaminants. What are the possible contaminants that would result in these products? One of them for the delta 8 conversion is the isomers of the cannabinoids. THC itself has 30 isomers, most of which are not found in nature, most of which have not been studied for safety or efficacy. There's also isomers of CBD, which are not found in nature. Those could also be part of this resulting mixture. Several of the isomers are produced in the synthetic reaction when the CBD is isomerized to THC, so we see this whole mixture of that. And the question is are these isomers a health risk? The first time that I saw Delta 10 gummies submitted to our lab, my first thought was, this is cool. People are thinking outside the box. This is really forward thinking. The next thing I did is I went to the literature and tried to find out what's the toxicity of Delta 10? What do we know about it? How does the body metabolize it? Do you know what information there is? None. At least that I could find. So here we're making products that we don't know anything about, and we're distributing them to consumers. In addition to those isomers, we see other synthetic reaction byproducts. They're cannabinoid-like. They might be chlorinated. They have other molecular functional groups on them, but they're not necessarily cannabinoids. But again, we don't know anything about these. We have bits and pieces of information on some of these contaminants. We might be able to tell that it's chlorinated. And we're starting to put together this puzzle, but we're missing some pieces, so we don't have the full picture yet. And again, as these unknowns have not been studied, we don't know what the toxicity of those are. In addition, the condition to isomerize CBD to delta 8 requires some pretty toxic chemicals and organic solvents, which may end up in your resulting product. How in depth is each producer removing those from a finished product? That's a question that I can ask because we don't know. And the challenge is that none of these products, or most of them, are being tested for these other synthetic reaction reagents. Recently, within the last 2 or 3 weeks, both the FDA and the CDC came out with some warning letters on Delta 8. The FDA had 5 things you should know about it. Number 1, Delta 8 products have not been evaluated or approved by the FDA for safe use and may be marketed in ways that put the public health at risk. Number 2, Delta 8 products often involve use of potentially harmful chemicals to create the concentrations of Delta 8 contaminated claimed in the marketplace. So that's what I was talking about in my last slide. The FDA has received adverse event reports involving Delta 8 THC containing products. They're not saying that these adverse events are from Delta 8, they're from the products themselves, so we can identify what the cause was. Delta 8 has psychoactive intoxicating effects. Delta 8 THC products should be kept out of the reach of children and pets. As for adverse events, there's been over 600, I think 660 some. 41% of them were unintended exposure, where people thought they were consuming a hemp product. Oftentimes when you see these in a vape shop, they'll say Delta 8, and for most consumers that's just a triangle 8. They don't know what the Delta is. But it doesn't really tell the consumer that it's a THC product oftentimes. 39% involved consumption of minors. 18% required hospitalization, including children needing to go to the ICU. So these are everywhere, they're available everywhere, and easily purchased by minors. The CDC also put out some publication listing some of the effects of the Delta 8 THC intoxication, lethargy, slurred speech, low blood pressure. A lot of these would be very similar to what you may see listed for a Delta 9 THC product. In addition to those big organizations, we also have public statements from several of the industry groups that might be interested in hemp or hemp production. The United States Pharmacopoeia They established standards for the pharmaceutical, nutraceutical world. They issued a statement that said the prevalence of synthetically derived delta-8 THC raises safety and quality concerns related to both identity and purity, given the unknown and untested nature of the synthetic analogs and the remaining compounds. The American Herbal Products Association deal with establishing safety and quality for herbal products. They discourage the marketing of goods for consumption by any route that consists of or contain any amount of artificial cannabinoids, which is any cannabinoid compound that interacts with cannabinoid receptors, but whose molecular structure is not found in nature. So again, delta-10, delta-6A, 10A, these are not found in nature. The U.S. Hemp Authority, they will not certify hemp products that are marketed for their intoxicating effects. This includes delta-8 THC. Again, the U.S. Hemp Authority has been very active in legislation, and they are concerned that delta-8 is an intoxicating component of hemp. They might lose legislative ground in the progress that we've made. The Hemp Industry Association has a little bit more favorable approach. They believe that prohibition is a failed concept and should not be applied to delta-8 THC or other hemp-derived cannabinoids. They advocate for the safe delta-8 THC product and production methods, and they encourage the FDA to regulate delta-8 THC and other hemp compounds, including CBD. So let's get into the chemistry. THC is a very complicated molecule. It actually has 30 different isomers, structural isomers as well as stereoisomers. There's a lot of different names for the delta-9, depending on what level of chemistry you are. Most people just call it D9. But basically, the D or the delta establishes where the double bond is in this ring. And so delta-9 THC, as we normally know it and love it, it has the double bond there. As we move the double bond around, we can see that these structural isomers, the delta-8, delta-7, delta-6A7, delta-6A, 10A, delta-10, and then delta-9, 11, also known as exo-THC. So those are the structural isomers that may result when you're converting CBD into THC. How do you know which THC you're going to get? It's even more complicated when you consider stereoisomers. These two carbons on this ring here are considered chiral centers, making this a chiral molecule. A chiral molecule has some meaning. When we draw hydrogens in this fashion, this is coming out of the page, this is going into the page. So the hydrogens are positioned like this. And that is the naturally occurring formation of delta-9. But if you're not restricting yourself to naturally forming delta-9 and look at synthetically formed delta-9, you can envision a conformation where the two hydrogens are up in the air, one where they're both down by the side, and the other one where they are reversed up and down. So those are all of the four stereoisomers of delta-9 THC. It gets even more complicated because some of those stereoisomers are also enantiomers, which is a special class of stereoisomers. It's basically defined as the non-superimposable mirror images. So if you were to take this structure and rotate it around, this is a mirror plane here. These are non-superimposable mirror images. And so these would be considered chiral compounds. Chirality is really important for our life. It's found extensively in biological systems, DNA, proteins, peptides, sugars, and yes, terpenes and cannabinoids as well are chiral. Many chiral compounds exhibit different biological responses. Here's a good example, carvone, which is a terpene. There's both an R and an S, or a right and a left-handed version of it. You can see that one has the hydrogen oriented up and one has it down. If I flip that around so that I can see the non-superimposable mirror images, you can see that these chiral centers are what make this a chiral molecule. The human body sees these differently. In one case, it sees it and it tastes and smells like spearmint, and in the other form, it tastes and smells like caraway because it's interacting in a chiral fashion with your tongue or your nose or your receptors. So drug response is dependent on these three-dimensional structures as well, and so it can be altered by stereochemical asymmetry. An example of how important this is the drug example thalidomide. Thalidomide was a drug that was manufactured in Germany in the late 50s. It was promoted for the treatment of anxiety, sleep disorders, tension, and morning sickness. It is a chiral molecule, so it has a right and a left-handed version. The right-handed version had sedative effects that was very useful in treatment of morning sickness and other things. However, the left-handed version was teratogenic and led to many deaths and birth defects. Approximately 40% of the children affected died at the time of birth. Those that survived had horrible deformities. And so this is how important chirality is to us. One chiral form was useful in treatment, the other caused these horrible birth defects. So when I raise concern about these other structures, it's because there is some historical basis for that concern. The isomerization from CBD into THC is not new. It was published in 1966 by Goni and Mishulam. It was further patented, but basically it's an acid-catalyzed isomerization and ring closure to form either the delta 8 or the delta 9. However, when you do so, you might get some of the other reactions occurring at the same time, depending on where this ring is when it's closing, which isomer you might get. In addition, delta 9 can also be isomerized to delta 8, and we actually see some regulated producers who have the licensed authority to produce delta 9 products also creating delta 8 products. Why would they do this? Maybe it's because of just consumer demand. There's a huge demand for delta 8 products. Testing these products as a laboratory is really challenging. Oftentimes, most laboratories are using LC-UV for the analysis of cannabinoids. And so many of the cannabinoids that we see have very similar UV absorbance, and sometimes these peaks don't match up with the retention time. In most of my samples, I will show you the actual reference standards that we use for identifying peaks. Those reference standards are created based on the cannabinoids that would be present in the cannabis plant. So when we look at the delta 9 reference standard, it's going to be the single stereoisomer that's found in cannabis. But as I look at the UV absorbance of those different peaks to try and help me identify them, it doesn't help much. Here's two unknowns that have the same UV absorbance as the exo-THC, the delta 9, the delta 8, and even similar to CBL. So looking at the UV absorbance alone is not helpful. If I look at some of the other cannabinoids, the delta 10s have very similar delta 6A, 10A. You can see a little bit of difference between the UV absorbance, and so that can be used as a tool to help us identify some of these compounds. Again, the red chromatogram on the bottom is reference standards, and the black chromatogram on top is what we would typically see. And you can see that the actual peak maxima do not meet the same retention time as the reference standard. And so oftentimes, laboratories will say, well, that's not delta 9, because if it was delta 9, it would be over here. And so those go uncharacterized as delta 9. The thing to remember, though, is that this black chromatogram, what we actually see and measure, represents a superposition of all the signals that we see. And so if it's actually a much more complicated mixture of components, it may not meet up exactly with the reference standards. And so there might be other delta 9 that when combined with it shift the peak. So oftentimes the presence of these unknown peaks is omitted from laboratory reports. Going to more expensive tools or instruments, we can go to LCMS. THC isomers are isobaric, which means they have the same molecular mass. So looking at the mass of CBD or THC, delta 9 THC, delta 8 THC, they all have the same molecular mass, and so it's very difficult to get any additional information by LCMS for those compounds. Going to GCMS, there's a little bit more information available. As we use mass spec with gas chromatography, we can actually get some fragmentation patterns of the ion that will help us identify those, because the compounds fragment in a very reproducible fashion. And most of those compounds can be searched against a NIST library database. That's the National Institutes of Standards and Technology. So as I try and identify these, as I start looking at delta 9, this is the fragmentation pattern for delta 9, delta 8, delta 10, delta 6A, 10A. The reason I'm showing these particular ones is because these are the standards that are most readily available. I have not been able to find delta 7 or delta 6A, 7. So we do what we can with what we have available. But what you'll notice is that they all have the same parent mass of 314. You can see that maybe delta 10 and delta 9 have very similar fragmentation, and that alone may not help us identify, but they have different retention times, so that's not a problem. But 6A, 10A, and delta 10 have very similar retention times, and I can, with a fragmentation pattern, distinguish between those. And so as I start applying that knowledge to the GC chromatogram that I showed, I can see that none of these three fragmentation patterns match any of the THC standards that I have. They do all have the same parent mass of 314, but that could be another isomer of THC, could be another isomer of CBD, it's hard to say. As I look at the other end of the chromatogram, I can see by these fragmentation patterns that again they have the same parent mass of 314, but these fragmentation patterns actually help us identify which compounds those two peaks were from. And so we're starting to use that as part of our tool set. Here's an example of a CBD to THC finished product. This was actually labeled as a delta 10 gummy, as I said before, but as you can see from the chromatography, there's very little delta 10 in it. It's more delta 6A, 10A. This is an example of a vape cartridge. We've seen thousands of these samples, and for the most part, they're all going to look the same. But you see all these things that are marked in red are compounds that we do not find coming from cannabis. They're not found in nature. When I look at the fragmentation patterns for some of these other unknowns and search those against the NIST library database, I get no hits, which means there's no ability for me to use that as a tool to identify those. We're also seeing them from delta 9 products. This was reported to me as a delta 9 THC distillate. And in this particular sample, about 23% of the chromatographic signal is not from naturally occurring compounds. Most of it is, again, at delta 6A, 10A. How that happened in a distillation, I'm not sure what their setup is. I don't know how that came to be. But it means that there's a concern for these types of compounds, not just from CBD to THC conversions, but just in general about cannabinoid safety, understanding what we're working with. So I call it the Delta 8, Delta X wheel of chance. Commercially available Delta 8 products represent complex synthetic mixtures. Many laboratories are not reporting these unknown constituents, so the certificates of analyses are often unreliable. They'll tell you that it's 82% Delta 8, but they won't tell you what the remaining constituents are. And because of that, producers often don't know what they're producing or what they're putting into the consumer stream. Without that regulatory oversight, consumers have no idea of what they're getting. So when I think about these, these are some points to ponder. Are these unknown compounds, are they going to do damage because the body doesn't know how to metabolize them? What happens to them in the liver? Will they cause cancer with repeated long-term exposure? Might some compounds cause birth defects if consumed during pregnancy? Will some of these compounds interact with other medications that a patient might be taking? Will the use of these products trigger a positive drug test? These are all questions that I don't have any answers to, and I don't think I've met anyone who does.
Wow.
We're certainly looking for them.
Oh my gosh.
In terms of drug testing, again, Delta 9 and Delta 8 are isobaric, which means they have the same molecular mass, and so a lot of the metabolites will also have the same molecular mass, and so whether or not consumption of a Delta 8 product will trigger a positive drug test might depend on how much you consume, the route of administration, what type of test is used, etc. When I think about these contaminants being low level, we keep asking ourselves aloud is, how much is too much? At what point am I concerned? So I look to the pharmaceutical industry and the FDA, put out some guidance that says that there should be a 0.1% threshold above which isolation and characterization of individual impurities should apply to chemically synthesized drug substances. Delta 8 is a chemically synthesized drug substance, so they would claim that anything above 0.1 should be studied. The United States Pharmacopoeia, with the International Council for Harmonization, has slightly different standards, but reporting above 0.05, which means if it's greater than 0.05 in a sample, it should be at least reported or acknowledged. If it's greater than 0.1%, it should be identified. And if it's greater than 0.15%, it should not only be identified, but studied for biological safety. I'm also asked quite often about CBN. Do I have concerns about CBN? Their processes are somewhat similar in that you're forcing the degradation of maybe delta 9 THC to CBN or CBD to CBN. But I don't have quite the same concerns. Yes, there's concerns, but we typically don't see the same level of impurities. This is just a chromatogram that I pulled out of the lab, and you see there's little blips of some impurities there, but it's really low level. Why is that? Maybe it's production of CBD. CBN is, because it's an acceleration of a natural process, maybe that's why we don't see these impurities. Could be that there's less competing synthetic pathways. CBN is also recrystallized, which helps purify. So maybe the recrystallization itself removes some of the synthetic contaminants. Recrystallization is not a feasible pathway for delta 8 products. So what's next? Now that we've got our foot cracked open the door for synthetics, we see THCP, which has a little bit longer tail on it, a 7-carbon tail. Hexahydrocannabinol has the double bond removed. And then the THC acetate, both delta 9 and delta 8 acetate. Delta 9 THC acetate has been known for some time. Delta 8 THC acetate products are hitting the market pretty wildly right now. Acetylation itself is yet another chemical reaction, another synthesis. It involves the addition of an acetyl group to a molecule in a position in which a hydroxy group is attached. It decreases the molecular polarity and thus increases the lipid solubility and membrane permeability. So that's used extensively in the pharmaceutical realm to enable such esters to cross blood-brain barriers and placenta. So if you go back, remember that the aspirin, salicylic acid, here's the example, salicylic acid is acetylated to make what is commercially available as aspirin. The acetylation is also used to convert morphine to heroin. So it's basically these hydroxyl groups are being acetylated to create heroin. So these are pretty standard reactions. Similarly, if you take THC, and this can be a delta 9, delta 8, and you acetylate it, you're going to add the acetyl group right on the what was once the hydroxy group. The THC acetate, because it's more crosses the blood-brain barrier easier, it has more psychoactivity than delta 9 THC itself. The reagents required to do the acetylation, they're highly corrosive. Breathing acetic anhydride can irritate the nose, throat, and mouth. High concentrations can cause severe lung damage in reaction with water. Acetyl chloride produces toxic and corrosive fumes, including hydrogen chloride and phosgene. Reacts with moisture in the air. Other ingredients or reagents include hexane and sulfuric acid. So these are all compounds that you don't want any residual of those in your final vape cartridge. This is an example of a THC acetate vape cartridge. As you can see, it's primarily was the delta 8 acetate. But again, all of these unknown compounds would normally be seen down here, but now that they've been acetylated, my guess is that those are all acetylated contaminants, which means they're even more potent at whatever they do, whether that's an efficacy response or a toxicity response. There's a path forward. Everything I've said has been pretty negative about delta 8, but I truly believe that delta 8 has a place in therapeutics. It has a place in recreational use. And so the path forward would be very similar to what is done in the pharmaceutical industry, where the synthetic reaction mixtures would then be purified or isolated. And there are chromatographic methods available for this. It facilitates the isolation and purification of the individual compounds. So not only do I want to purify the delta-8, I also want to purify the other constituents so that we can study these. Find out if they're toxic. Maybe these are novel compounds that have some great efficacy on their own. What are they? There's different ways to do that. One of the ways is supercritical fluids, CO2, preparative scale, SFC. SFC is a normal phase technique. It uses carbon dioxide as the primary mobile phase for the separation. CO2 is nice to work with because it's chemically inert, non-flammable solvent, and it can be recycled. In addition, it's really easy to remove it from your final product. As you collect your final